He has made sustained contributions to teaching, research, postgraduate training, academic leadership, and capacity development in agronomy, plant breeding, crop improvement, and agricultural biotechnology in Nigeria. Your Excellencies, very important personalities here present, consummate professors and academics, affable staff and students of the University of Abuja, family members, friends and well-wishers of the inaugural lecturer, gentlemen of the press, distinguished ladies and gentlemen, with a deep sense of appreciation and a standing ovation, please join me, Agoma Awaitoma, in welcoming to this lectern a dexterous agronomist, astute scientist, consummate professional, outstanding intellectual, and distinguished scholar, Professor Bashir Omolaran Bello, as he comes to deliver his inaugural lecture. You're welcome, sir. Congratulations.
In the name of Allah, the most beneficent, the most merciful, the Vice Chancellor, Deputy Vice Chancellors, the Acting Registrar, and other principals, officers of the university, Provost, College of Earth Sciences, Dean, Faculty of Agriculture, Deans of other faculties, postgraduate school, and student affairs, directors of various units, professors and other members of the Senate, head of the Department of Agronomy, heads of other departments, other members of the academia, administrative and technical staff, distinguished invited guests and friends, members of my family, nuclear and extended, gentlemen of the print and print electronic media, great University of Abuja students, great Nigerian students, distinguished ladies and gentlemen, Mr. Vice Chancellor, distinguished colleagues, honored guests, ladies and gentlemen, I give glory to Almighty Allah for his guidance and mercy and abundant blessings. I'm deeply honored to deliver the 51st Inaugural Lecture of the University of Abuja, the 5th from the Faculty of Agriculture, and the 1st from the Department of Agronomy. Role of plant breeding in enhancing climate resilience, improving quality, and strengthening sustainable food systems is my lecture focus, titled Plant Breeding for Climate Resilience and Food Security, from Genes to Sustainable Food System. My personal journey began on my father's farm and was shaped by my academic training, research, and professional experience. The key message: plant breeding bridges genes and sustainable food system, providing pathways to climatic resilience, improved nutrition, and food security. Food comprises of nutritional support, such as carbohydrates, protein, fats, water to human body. By 2050, the world population is projected to 10 billion population. And there is a problem of global warming, where greenhouse gases emissions were emitted to the atmosphere due to fossil fuels, afforestation, and among others. Climate change is a long-time changes in Earth climate and increases drought, floods, pests and diseases, thereby strengthening crop productivity. Climate resilient crops withstand and adapt and recover from the adverse effects of climate variability. Food security is a fundamental requirement of human well-being. Economic development, social stability is built on 4 interrelated pillars. These are availability, accessibility, utilization, and stability, which are for sustainable food systems. The key message here is that climate change threatens food security, necessitating the development of climate-resilient crop variety to sustain food systems. Understanding plant breeding. Plant breeding is an art and a science that exploits genetic variation to develop high-yield, climate-resilient, nutritionally enhanced, and farmer-preferred crop varieties. The act of plant breeding relies on breeders' experience, intuition, creativity, and field observation to identify superior plants and desirable traits for crop improvement. The science of plant breeding applies genetics, genomics, biotechnology, and other advanced tools to understand inheritance and accelerate genetic improvement. Successful plant breeding combines breeder expertise with modern technologies to improve selection accuracy, breeding efficiency, and genetic gain. The key message is that plant breeding transforms genetic diversity into improved varieties that strengthen climate resilience, food security, and sustainable food system. These are the objectives of plant breeding. 1, increase in yield, develop varieties of high productivity, such as high-yielding maize and rice, improve disease resistance, reduce losses from diseases such as southern corn rust, covularia, maize streaks, enhance pest resistance, developing variety that withstand insect attacks such as fall armyworm and stem borer, improve abiotic stress tolerance, that is enhanced tolerance to drought, heat, salinity, low soil fertility. That's why drought-tolerant varieties are being researched on. Other objectives are increase in essential nutrients in crops such as vitamin A maize, orange flesh sweet potato, promote early maturity, development of varieties that mature faster, such as extra early maize variety my research dwelled in. Also improve development of cultivars suited for adverse environmental, such as savanna adapted maize varieties. Increase resource use efficiency, improve water and nitrogen use efficiency in maize. Strengthening climate resilience, develop varieties that maintain yield under climate variety, such as drought-tolerant hybrid. Enhance plant architecture and lodging resistance. Also, the last, meeting and consumer preference, that is improve quality, taste, and processing characteristics. Historical stages of plant breeding. Plant breeding started from selection of use of plants from the wild since 10,000 BC to 18,065. From 1865 to 1980, we have the father of genetics, which is Mendelian, where he discovered dominance and recessive traits. From 1980 to 2015, plant breeding has transformed to the use of deoxynitric acid markers and molecular tools, then tissue culture and biotechnology. Plant breeding, tissue culture. Tissue culture is the production of disease-free uniform plants. And this is usually done in the laboratory under controlled environment with AC and avoidance of uncontaminated materials. Based on my picture, the explant from leaves, stem, even another branches can be used in culture because of something potency, that is ability to generate from the cells. In the lab, explant will be cut and it will be subjected to using suitable disinfectant under aseptic condition. Then it will now be inoculated onto a sterile nutrient culture medium. For example, molasses. Next is that now be incubated under controlled light, temperature, and humidity to produce shoots and root formation. Next is subculture as necessary to multiply roots. And the last will generate the plant in the soil after hardening up under no-stream conditions.
my research